at chapter 11, part 2 of 2, still of the muscular tissue, but we're going to dive into muscle metabolism. I kind of like this area. It's pretty nice. Hopefully it's not too difficult to understand. You know what to do. If it is, click out the major important parts and put it into YouTube and see if there's any extra videos to help you understand. I'm going to see if I can post some for you. But if the ones I post don't help, then just check again and see if there's anything else, or just read and highlight and take your time and understand everything. Okay, so talk about muscle metabolism. All muscle contraction, it depends on ATP. Now, and the supply of ATP, it depends on the availability of oxygen and organic energy sources such as glucose and, of course, our fatty acids. Alright, so there are two main pathways of ATP synthesis. And what are they? Anaerobic fermentation and aerobic fermentation. Now, during the remaining part, we're going to deal with the different mechanisms that produce ATP depending on the duration. We're looking at the time frame. Either it's 10 seconds, 1 hour, 2 hours. That's what we're looking at. How the ATP is produced during that time frame. So when we look at anaerobic respiration or fermentation, let's get a definition of these two first. When we talk about anaerobic fermentation, respiration, we're looking at respiration without oxygen. Now, the process uses respiration, electron transport chain, but does not use oxygen as the electron acceptor. So oxygen is not needed in order for this process to begin. When it comes to aerobic respiration, this is the respiration that needs oxygen to continue. Okay, so the anaerobic fermentation, it allows the cell to produce ATP without the need for oxygen, like I mentioned, but the ATP yield is limited. Okay, and the process generates lactic acid, which can contribute to muscle fatigue. Now, with aerobic respiration, it produces more ATP and no lactic acid, but it requires a continuous supply of oxygen. Now, in a resting muscle, most ATP is generated by the aerobic respiration of the fatty acids. Now, when we look at aerobic respiration, the amount of oxygen, the amount of energy that's released, it is lower than, it's usually between 2 to 36 ATP molecules that are produced. But during the aerobic respiration, you have about 36 to 38 ATPs that are produced. Okay. Now, when we look at this particular diagram here, or this figure, we notice at 0, which is our duration, at 0 duration, we have aerobic respiration using oxygen from our myoglobin. So we're using oxygen that's already within the muscles. That's just to get us in our start position, not much movement. And then when the gun goes off and you hear start, that start gun goes off, and you're sprinting for about 10 seconds, we have it's called phosphagen system that comes into play to help with helping you move to complete those 10 seconds. And as time increases, we have the glycogen lactic acid system, which is still in the anaerobic system. This is about 25 seconds or so, and then from 40 and onward, we have the aerobic respiration, which is supported by our heart and our lungs, which is our cardiopulmonary function. Alright, so when we look at, let's say like a 100-meter dash, that's considered to be a short, intense type of race. So if you've watched track, I love track and field. So if you watch track and you've seen Usain Bolt or Alison Felix run that 100-meter dash, you see that they are explosive, and it's a very short race, lasting about maybe 9 seconds now. I think that's the world war, the record now, less than 9 seconds or so. In this particular exercise, a short, intense exercise, we get the oxygen from the myoglobin. So the muscle is going to borrow these phosphate groups from another molecule and then transfer them to ADP to form ATP. Now remember, ATP is adenine triphosphate. So it has 3 phosphates on this adenine molecule. ADP is 2 phosphates, adenine diphosphate. So it has 2 phosphates and on this adenine molecule. Just hoping you guys remember this. So what happens after it bars that phosphate group? We have the 2 enzyme systems that are going to control these transfers, and that is our myokinase, and our creatine kinase. Now the myokinase, they're going to transfer that phosphate group that was taken from the ADP, and they're going to transfer ADP from they're going to transfer the phosphate from one ADP to another, converting it to ATP. It's going to take a phosphate group from another ADP, and they're going to use that and combine it with ADP to make our ATP. So our creatine kinase, then, they're going to obtain a phosphate group from the creatine phosphate, our CP, and then donate it to ADP to make ATP. So here, these are the 2 enzymes that are helping to make ATP without the use of oxygen. The myokinase is going to use it from another ADP group, and then we have the creatine kinase. And the creatine kinase says they're going to obtain their phosphate group from creatine phosphate group to help with making more ADP. Okay, so ATP and the creatine kinase. Sorry, no, the ATP and the creatine phosphate. Sorry, they are considered to be the phosphogen system, and they provide nearly all of the energy used for short bursts of intense activity, such as sprinting for about 6 seconds or so. So we're looking like at a 6-second sprint, or no, 6-second sprint, maybe 50-yard dash. Maybe soon it'll be the 100-yard dash as well. Now, as the phosphogen system is exhausted, the muscles shift into this anaerobic fermentation for short-term energy until the cardiopulmonary function can catch up with the oxygen demand. So the longer we run is going to activate or is going to require oxygen, shifting us over to rely more on that aerobic system. Now during this period, muscles, they're going to obtain glucose from the blood from their own stored glycogen. Now the pathway from glycogen to lactic acid is called glycogen lactic acid system. And that produces enough ATP for 30 to 40 seconds of maximum activity. So here we have these two enzymes. So here we have ADP, here we have the kinase, the myokinase, and we have the creatine kinase. And what happens, in order for the energy to start off without using oxygen, we have the myosin kinase is going to take a phosphate group from ADP. So remember, it has two. So it will leave it with just one, called an adenine monophosphate. So this phosphate group is collected from the myokinase, and then it takes it over and combines it with another ADP that's going to help with making ATP. So that's two phosphates making three phosphates are ADP. Then our creatine kinase, they're going to take a phosphate from creatine phosphate. So creatine phosphate only has one phosphate, and creatine kinase is going to come and steal it, take it away, leaving it with just creatine. That phosphate group is carried over to an ADP, which is lingering around, to create ATP, and this is what's going to help with these short bursts, intense type of exercise or activity, I should say. All right, so when we look at the phosphagen system, which we already said is the ATP and the CP, or creatine phosphate, it's going to exhaust the muscles, and it's going to shift to anaerobic fermentation. So I already went over this part where the muscles are going to obtain the glucose from the blood, and if we have any stored up glycogen, they're going to use that too. Now, in the absence of oxygen, glycolysis is going to generate a net gain of 2 ATP for every glucose molecule consumed. Then that is going to convert that glucose to lactic acid. So our glycogen lactic acid system is the pathway from glycogen to lactic acid, and it produces enough ATP for 30 to 40 seconds of maximal activity. So it's basically running up the stairs quick, or running for your car to the front door. Just 30 seconds or so. Okay. Now when it comes to long-term energy, after 40 seconds or so, the respiratory and the cardiovascular system, they're going to deliver oxygen to the muscles fast enough for aerobic respiration to meet most of the ATP demand. So now we're in the aerobic respiration, where we need that oxygen, and this is just after 40 seconds. Now, aerobic respiration, it demands much more ATP and is a very effective means of meeting the ATP demands of prolonged exercise. Now, oxygen consumption, it rises from 3 to 4 minutes, and then levels off at a steady state in which ATP production keeps pace with the demand. So after it gets to 3 minutes, it's going to level off. So let me see if I can, I don't remember if I have it in one of the other slides, but we have our anaerobic respiration, where not much O2 is needed. Oh, let's see, right there, down here. Oops. Okay, get rid of that one. Alright, so here we have our graph. Here's our O2 consumption. O2. So here with our anaerobic respiration, at about 30 seconds or so. Yep, 30 seconds. What's going to happen? 30 to 40 seconds. I'll put 40. That's a 4. We have from 0. We have anaerobic respiration. When we get to that 40 seconds, we need oxygen, correct? So oxygen is going to increase. The demand is higher until we get to, I'll say, about 4 minutes. And then at that 4 minutes, it's going to level off. And that's all if the intensity has stayed the same. Okay, so that's you starting off on your jog. So you're going to run the 5K, which is a 3.3 or 3.6 mile run, 3.3 mile run. But you're gonna run that 5K. When the starter gun goes off, you hear you start off when your anaerobic fermentation or respiration. When you get to that 40 seconds, here you have the generating of ATP, more ATP, because you need oxygen in order to complete that race. After 4 minutes, if you're on that steady pace to complete that 5K, everything is steady. You have a steady amount of ATP produced and a steady amount of oxygen requirement in order for you to complete your race. Okay? Alright, so also in this area we have a little lactic acid. It's going to accumulate under that steady state conditions, and we have a depletion of the glycogen and the blood glucose, and together with the loss of fluid and electrolytes, this is through sweating, of course, the set limits on endurance and performance even. It sets limits on the endurance and the performance even when lactic acid does not occur. So everything's going to come to a halt at some point or time. And that's because you're going to deplete your glycogen and your glucose eventually. You're going to lose a lot of fluids and electrolytes. Of course, what's the body going to thrive on then? So at a point in time, your body's basically going to say, you know what, it's time to stop running, because I cannot provide you with enough ATP, glucose, glycogen, and we're losing so much fluid and electrolytes, so we have to stop. Otherwise, you are going to pass out, faint, and maybe die if taken to extreme measures. Alright, so I'm going to look at muscle fatigue. Muscle fatigue is the progressive weakness and the loss of contractility that results from prolonged use of the muscle. So, for example, is repeating squeezing a rubber ball. So it's holding a ball in your hand, just squeezing it, just say for a minute, and all of a sudden you want to continue on. I'm going to try to do it for maybe 2 or 3 or 5 minutes. Eventually your muscles are going to get weak and get tired, and you won't be able to squeeze that ball for a long period of time. Even holding a textbook, like the next example is holding a textbook out level to the floor. So holding a textbook and extending it out in front of you. Over time your muscles are going to be tired and weak. So fatigue is thought to result from ATP synthesis declines as the glycogen is consumed. So ATP is not being produced anymore, but our glycogen is being used. Okay. Place. Okay, so fatigue has multiple causes. We have potassium accumulation, where in which each action potential the potassium accumulates in the extracellular fluid. This is going to lower the membrane potential and make the muscle fibers less excitable. So remember, we need the potassium inside is where it's supposed to be, and sodium outside. But now we have a lot of calcium on the outside, which is not what we want. We're just going to throw off the voltage. We have negative outside on the extracellular fluid and positive on the intracellular fluid, and that's not what we want. Then we have ADP accumulation and phosphate accumulation. This is the growing pool of ADP plus the phosphate generated by ATP hydrolysis, and it slows down that cross-bridge cycling mechanism of the transaction. Then we have lactic acid accumulation. This is while it has long been thought that the lactic acid contributes to fatigue by lowering the pH in the muscle fibers, many now contend that the muscle fatigue is independent of lactic acid. Then we have fuel depletion. Depletion as muscle glycogen and glucose levels decline, less ATP is being synthesized, is being made. Then we have electrolyte loss. Loss of electrolytes through sweating can alter the electrolyte balance of the extracellular fluid and then reduce muscle excitability. And lastly, we have central fatigue. Let's see what's on this page. No. Yeah. Okay. Sorry. Then we have central fatigue, where exercising muscles are going to generate ammonia, which is absorbed by the brain and inhibits the motor neurons of the cerebrum. For this and other reasons not yet understood, the central nervous system produces less signal outcome to the skeletal muscles. Alright, so what is endurance? Endurance is the ability to maintain high intensity exercise for more than 4 to 5 minutes. So it's not just running a 10-mile race, but basically if you 6 minutes, 7 minutes, 10 minutes is basically endurance. Consistently though, at a high intensity, not just walking around the park with a phone in your hand. I've seen that many times. Walking around the track with a phone in your hand, talking on the phone, thinking you're doing some type of exercise. Yes, I'm working on my endurance, I want to get fit, whatever it is, but you're not doing anything. You're just walking, talking on the phone. But endurance, in order to increase the endurance, you need to go, your intensity needs to be high and be consistent for 4 to 5 minutes. Sorry, I went off a little bit. The maximum oxygen output, which is called our VO2 max, is the point at which the rate of oxygen consumed, the amount of oxygen that you bring in, reaches a plateau and does not increase further with an added workload. So you are consuming a lot of O2, but your workload has not changed. No, your workload has changed. You're bringing in you're consuming a lot of O2, and you reach a plateau, but it does not increase anymore, even though you are working harder. Okay, and it determines on a person's ability to maintain high intensity exercise for more than 4 to 5 minutes. Now, VO2 max is proportional to the body size, and it peaks at around age 20. It is usually greater in males than it is in females. It can be twice as greater in a trained endurance athlete as in a untrained athlete. So, I can get my little chart out. So, when you do a VO2 max, here's your VO2. And here's your just say intensity is over here. Now with a trained athlete, they will get to their VO2 max a lot quicker and sustain it. Now for a untrained individual, I need to change colors. The untrained individual take a longer time to get to that VO2 max, and eventually they'll level off. So here we have an endurance athlete that has started off with that 5K or 10K, and about 5 minutes into the race, or maybe 10 minutes into the race, they're at VO2 max. Steady pace, going strong, and here we are, untrained in that particular event. We are struggling just to keep up, panting, trying to get that VO2 max. But theirs will be a lot higher. Ours will level off right here. Sorry, it won't be that high. Let me erase that so you don't get confused. Our VO2 max will be a lot. It erased. Okay, let me get the color. Sorry. Our VO2 max will be a lot lower. We're not going to go as long as they're going to go, but our VO2 max will be a lot lower. Okay, so theirs always comes with training. You have to train for those type of races to increase that VO2 max. Okay, swimmers will also have a high VO2 max. Okay. Okay. Okay, so how do we beat the fatigue? How do we even get past fatigue? Oh, I forgot to mention, for a sedentary adult weighing approximately 160 pounds, their VO2 max would be about 35 milliliters per minute per this is my sedentary person, 35 milliliters per minute per kilogram. That's how VO2 is max. VO2 max is written milliliters per minute per kilogram. But our trained athlete, theirs would be 70 milliliters per minute per kilogram. Okay. Now, with excessive post-exercise oxygen consumption, which is EPOC, this is also known as oxygen deficit. Okay, I skipped over something. Let me get back to beating fatigue. Now when you take oral creatine, it's going to increase your creatine phosphate in the muscle tissue, and it's going to increase the speed of ATP regeneration. Because remember we talked about the creatine phosphate, how it's going to the creatine kinase will take a phosphate group from the creatine phosphate, and you take that phosphate group and add it to an ADP to make ATP. So this oral creatine is going to increase that process. Okay, and it's useful in burst type exercises such as weightlifting. You already know that steroids helps out as well, and that's banned, of course, with various sports, especially track and field. They've had a number of issues, especially baseball and all that. But risks are not well known. They say it causes muscle cramping, electrolyte imbalance, dehydration, water retention, of course, a stroke. And also, they say it causes kidney disease from overloading the kidney with metabolite creatine, which is definitely true. You know, your kidneys are that filtering system in the body. Then we have the carbohydrate loading. That's eating a large amount or a heavy amount of carbohydrates. Your pasta, your breads, consuming that, so you're gonna overload on the body. Usually they might do that I think a day or so, the day before a race, so that you're packing the body with glycogen. Because when you consume the carbohydrates and it's broken down, it's the electrolytes, not the electrolytes, sorry, it's the glycogen. Glucose is used, but then if that's used and you have some left over, it's stored as glycogen in the liver and of course in the muscle cells. So they want to make sure they pack up their muscle cells with a lot of glycogen, so that they have enough to finish that race, especially those endurance races. So extra glycogen is hydrophilic, and it adds 2.7 grams of water per gram of glycogen. So athletes feel less sense of heaviness, overweights, and it benefits of extra available glycogen. So when we talk about heavy breathing, it continues after strenuous exercise, and we have EPOC, which is our excessive post-exercise oxygen consumption, also known as the oxygen deficit. And it's the difference between the elevated rate of oxygen consumption following an exercise and the rate of oxygen consumption at rest. Excess oxygen consumption after strenuous exercise occurs, in part for the following reasons. If oxygen is required to anaerobically synthesize ATP, and some of that ATP is used to generate that creatine phosphate. Okay. Now, a small amount of oxygen it serves to oxygenate the myoglobin. Now, the liver, it consumes oxygen and disposing of the lactic acid produced during exercise. Now, exercise raises the body temperature and overall metabolic rate, which consumes more oxygen. Alright, so let's look at our muscle fiber classes, physiological classes. Hopefully you guys have heard of fast twitch, slow twitch. Okay, so when we look at the slow twitches are SO, they're also called slow twitch, they're red, or they're called type 1 fibers. They are the ones that you see in the endurance runners. They have a high fatigue resistance, so they're not going to fatigue fast. And that's to make sure that they're going to support the body and, of course, maintain the posture as well as the cardiovascular system for this long race. So this is a Kenyan Olympic runner who's been winning, who won at the Olympics 2000. Well, this is the last Olympics. And he is an example that will have this low twitch fibers. Okay, and they will have an abundance of mitochondria, an abundance of myoglobin and capillaries, and of course theirs will be deep red in color. So here's an example not of the slow twitch, but just of the various colors. Here's our dark red. They will have a multitude of dark red muscle fibers full of oxygen. That's why they're that color. Okay. Of course, you have a few white, which is fine, but the majority of this actual slide would have been dark red muscle fibers that you would have seen. Okay. Then we have our fast twitch fibers. They're white. They're considered to be white, and they are type 2 fibers, and they are adapted for quick responses, such as those that needed to control eye and hand movements. They have an extensive SR, or sarcoplasmic reticulum, with fast release and the reabsorption of calcium, and a form of myosin ATPase, and a fast ATP hydrolysis. So they're going to break down the ATP a lot faster. And the cross bridge cycling. Okay, and then also with these type 2 fibers, they depend on glycolysis and anaerobic fermentation. We're talking about no oxygen, which will be good for a quicker but less efficient production of ATP than aerobic respiration, which is supported by the high concentration of glycogen. Now, the fast glycolyc fibers, they have fewer mitochondria and are thicker than the slow twitched or slow oxygenated fibers. And they also have less myoglobin, causing them to be relatively pale and are therefore called white fibers. So let me go back to this other page. The fast twitch, they will have more of these pale looking color fibers. So I'll usually with this lighter pink in this particular slide. I mean, we do have those that are fast twitch A and B, um, A and B, and usually those are the ones that you'll see with this type of slide here. We have a combination of, they'll be pale, I mean a lighter, there'll be a pink color. So this will be a pale pink, this is a dark pink, this will be a regular pink color. That'll be your, like your fast twitch 2A and B, and those are the ones that can run the long distance races as well as the short distance races. Just as this is Allison Felix. She runs the 400 meters, which is endurance race, as well as the 100 meters. Of course, this is the United States. They won the 4x1. This is the 4x1 team, and they did break the world record. As you can tell, I do love track. And here is the Jamaican team with just two runners, Usain Bolt and Blake. They run the 100 meters 4x1, also the 200 meters as well. Okay, drifting off, I'm sorry. Okay, so we also forgot to mention, when we talk about these fast twitch our fast twitch fibers, I didn't mention about the A. They are oxidative and and fatigue resistant, and their fibers are kind of like unknown to humans. Now, each skeletal muscle is composed of both SO, which is our slow oxidative, and our fast glycolic fibers, but the proportion, it differs according to the function of the muscle. Now, the fiber types can be distinguished and with stains of oxidative or glycolic enzymes. Now, muscles composed mainly of SO fibers, which is our slow oxidative, our F1, our type 1. They are called red muscles, and those composed mainly of fast glycolic fibers. And those with fast glycolytic fibers, they are called white muscles. Now, the proportions differ even in a single muscle in people with different types and levels of physical activity. Now, of course, heredity is going to play a role in which a person is either a born sprinter or a born marathoner. So for Usain Bolt, definitely he is a born sprinter. And with him, of course, his gastrocnemius, which is his calf muscles, they're going to be predominantly fast twitch. So you see, this would be if they ever did a sample of his muscle cells in his calf, this will be an example of what it will look like. A lot of fast twitch fibers here, and slow. Okay, but if you have a long distance runner, such as the other Kenyan I showed you, his calf muscles will have a lot more dark pink, within it, and fewer pink. But with Allison Felix, for hers would be a mixture of both. Okay. Alright, so humans have far more muscular strength than is normally used, which is true, and muscles can generate more tension than the bones and tendons can withstand. Now, muscular strength depends on anatomical and physiological factors. We have muscular strength is primarily determined by the muscle size, the fascicle arrangement, and of course the size of the motor units. Alright, so the muscular strength depends on the basically the muscle size. The muscle can exert a tension of 3 or 4 kilograms per centimeter square of the cross-sectional area. When we look at the fascicle arrangement, and we talked about the pinnate, The pinnate are stronger than the parallel, and the parallel are stronger than the circular. So I hope you guys remember the pinnate, the type of the pinnate is shaped more like this. This is the muscle fiber, where it looks like a fan. Has one end where the tendons are, where the tendon is located, and then it branches, it spreads out like a fan. That's your pinnate. Then we have the parallel, which is just straight, and then we have the circular, which is like this. This is your muscles. So you're looking at around the eyes for this one, or any opening. This one you may be looking at the sartorius muscle or the gracilis muscle in the thigh, and this one you look at the pectoralis major or minor. Okay. Alright, and then we have the size of the unit. Now, the larger the unit, the larger the motor unit, the larger or the stronger the contraction. Okay. Then we have multiple motor unit summation, of course dealing with recruitment. Now when stronger contraction is required, the nervous system activates more motor units. Then we have temporal summation, temporal recruitment. We have nerve impulses. They usually arrive at the muscle in a series of closely spaced action potentials. So the action potentials, they're going to cause a stronger contraction. Then the length and tension relationship that contributes in that a muscle resting at optimum length can contract more forcefully. And then resting muscles, they contract more strongly than fatigued ones. That's basically saying when you rest the muscles, you take a day or two between your exercises, depending on what muscle group you're working on. Like if I'm working on, say biceps today, it doesn't going ahead and working on biceps tomorrow is not gonna be as effective as waiting 2 days, or having one rest day before you going back, or 2 rest days before going back to working on those biceps. Okay. Alright, then we have resistant training. Nothing wrong with resistant training. Resistance exercise such as weightlifting can stimulate muscle growth even if only done a few minutes at a time, a few times a week. If you do 30 minutes today, you can do another few 30 minutes 2 days time. Every 2 days, do something in type of resistant training. Now, growth results primarily from the cellular enlargement, not the cellular division. Now, myofibrils, they grow thicker and split longitudinally when they reach a certain size. Now, muscle fibers are incapable of mitosis, but of course, some evidence it indicates that they can also split longitudinally as they enlarge. We look at endurance runners or endurance exercise, such as jogging and swimming, cycling, like the Tour de France. It improves the fatigue resistance of the muscles by enhancing the delivery and the use of oxygen. So you'll be able to handle the amount of oxygen that's coming in to reduce your muscle fatigue time frame. If you're used to just running for 1 minute and you're tired, if you constantly do it on a regular basis, you'll be able to run a lot longer, or swim a lot longer, cycle a lot longer. Maybe get up to 5 minutes, 10 minutes, and maybe eventually to 30, 40 minutes. Okay. So it's going to improve your fatigue resistance. Okay. We have slow twitch fibers. They're going to produce more mitochondria and more glycogen and acquire a greater density of blood capillaries with conditioning, like I just said. Now, this form of Excite also improves skeletal strength and increases red blood cell count, oxygen transport capacity, and it enhances the cardiovascular, respiratory, and the nervous system function. Why am I stressing it? We have to get up and do something. Get up and do some type of aerobic exercise. Alright, so let's look at the cardiac and smooth muscle. Yes, we just went over the skeletal muscle. There was a lot involved. But now we're going to hit the cardiac muscle and the smooth muscle, the very last two. So let's look at the cardiac muscle just a little bit. Here's a nice slide on a cardiac muscle. I'm showing these so when I start talking about it, you have a general idea of what it's supposed to look like. Okay. Here's our cardiac muscle. We have branch cardiac cells, which we have in here. We have these intercalated discs, which over here you'll see them right here. Here's our intercalated discs. That's a better view. What they look like. Somewhere here. Those are intercalated discs. And then we have our nuclei of the cardiac cell. These dark pink cylinder-looking structures. Okay. Here's another view of our intercalated discs. The same thing. Okay. Alright, so we look at the cardiac muscle. It's limited to the heart where it functions to pump blood. The properties of the cardiac muscle, we have 5 properties. The first one deals with it must contract with a general rhythm. The muscle must function in sleep and wakefulness, while we're asleep and awake. It is highly resistant to fatigue, cannot get tired. The cells of a given heart chamber must contract in unison. And each contraction must last long enough to expel blood from the chamber. So those are the properties of the cardiac muscle. Now the cardiac muscle is striated, like the skeletal muscle, but the cardiocytes are shorter and thicker. Now the cardiocytes, those are basically the cardiomyocytes. Okay. Now each myocyte is joined to several others at its ends through the linkage called intercalated discs, which I showed you what those intercalated discs are. Now these discs, they appear as thick dark lines and stained tissue sections. So let me go back just a little bit to remind you. Here they are. Your intercalated discs. Okay. Now intercalated disc has electrical gap junctions that allow each myocyte to stimulate neighbors and mechanical junctions to hold the myocytes together. Now the sarcoplasmic reticulum of the cardiocytes is less developed than in the skeletal muscle, but has larger T tubules. Okay, so let me move forward again to the slide that I was on, if I can remember where it is. Okay, I'll stop right here. Alright, so when we have damage to the cardiac muscle, it is repaired by fibrosis. Now, although mitosis has been detected in cardiac myocytes following heart attacks, it does not produce a significant amount of regenerated muscle. Cardiac muscle contains a built-in pacifier that rhythmically sets up a wave of electrical excitation. Now, cardiac muscle is said to be autorhythmic because of the ability to contract rhythmically and independently. The heart does not receive fibers from the autonomic nervous system. Sorry, it does. It does receive fibers from the autonomic nervous system that can increase or decrease heart rate and contraction strength. Cardiac muscle does not exhibit quick twitches, but contains tension for 200 to 250 milliseconds. Now, cardiac muscle uses aerobic respiration almost exclusively. It is rich in myoglobin and glycogen and has large mitochondria that fill 25% of the cell. It is adaptive with respect to fuel, but vulnerable to lack of oxygen. Now, because little anaerobic fermentation takes place, the cardiac muscle is highly resistant to fatigue. Okay, so that's the cardiac muscle. So let me look at the smooth muscle. The smooth muscle myocytes are named smooth due to the lack of striations and are different in many ways from the myocytes of the skeletal muscle. Now, the smooth muscle is not always innervated, but when it is, the nerve supply is autonomic. The nerve fibers do not form neuromuscular junctions with the myocytes. Each fiber has about 20,000 bead-like swellings called where are we? Okay. Called varicocytes along its length. Alright, so each viscosity, it contains synaptic vesicles, which we talked about earlier, from which it releases these neurotransmitters. Now, the myocytes, they don't have motor end plates, but instead they have receptors for the neurotransmitters on their surfaces. Alright, so we have our Z discs. Now, our Z discs are absent, and in their place are protein plaques called dense bodies. And those are what you find on the inner plasma membrane, and then they disperse throughout the whole sarcoplasm. So let's take a look at the smooth muscle. We see that it's composed of myocytes with a fusiform shape. Now we see that there are no striations; they're completely smooth, and they're about these myocytes. They're about 30 to 200 micrometers in length, 5 to 10 micrometers in width at the middle, and they start to taper off at the end. And there's only one nucleus. Now the sarcoplasm is scanty, and there are no T tubules. Thick and thin filaments are both present, but they are not aligned and produce no various striations. Okay. An extensive cytoskeleton network of the intermediate filaments is associated with these dense bodies. We have the actin filaments that that attach to these intermediate filaments and the dense bodies. I'm sorry, let me go back. And so their movements is transferred to that sarcolemma. Now we have, go back here. Now we have two functional types of smooth muscle. We have the multi-unit and we have the single unit. Now with the multi-unit smooth muscle, it occurs in some large arteries and pulmonary air passages, the patellar erector muscles, the ones that are going to raise the hairs on your hand or even on your body, and muscles that control the iris. Now, its autonomic nerve innervation is similar to the motor nerve innervation of the skeletal muscle, in that the terminal branches of the nerve fiber, they synapse with individual myocytes and form a motor unit. The various causities are associated with a particular myocyte, and each myocyte responds independently of all the others. Now, a single unit or unitary smooth muscle is more widespread, occurring in most blood vessels and in the digestive, respiratory, urinary, and the reproductive tracts. It is also called the visceral muscle. Now, its name refers to the fact that the myocytes are electrically coupled to each other by gap junctions, so that cells directly stimulate each other and can contract as a unit. So here we have our various causities, which I was trying to pronounce. Our various causities. Here's our autonomic nerve fiber. And here's the multiple mitochondria that you find within it, and the synaptic vesicles that are going to hold your neurotransmitters. Okay. Let's see. Alright, so smooth muscle stimulation is achieved in a variety of ways. Most muscle is innervated by autonomic nerve fibers that can either stimulate the muscle contraction or hinder it. So let me go back to this diagram right here. So here we have an autonomic nerve fiber that's going to stimulate the muscle contraction, right? That we have used in the smooth muscle. Now these fibers, they're going to stimulate the smooth muscle receptors with either the acetylcholine or the norepinephrine. Those are the ones you're going to find within these vesicles. So the nerves have contrasting effects in different locations, such as stimulating the gastric motility or dilating the bronchioles of the lungs. Okay, so smooth muscle, they react to chemical stimuli like hormones, carbon dioxide, oxygen, nitric acid, oxide, and low pH and some other ones. Now, low temperature is going to induce the contraction of the smooth muscle in the areola and the scrotum, while warmth relaxes smooth muscles in the arteries of the skin. So smooth muscles of the stomach as well as the urinary bladder, they contract when they are stretched. Now, some single unit smooth muscles, especially in the stomach and the intestines, they have pacemaker cells that set off waves of contraction, and that is the autorhythmic. Now, contraction in smooth muscles is triggered by calcium, energized by ATP, and then it involves the sliding thin filaments over the thick filaments. And this is called the mechanism of the excitation contraction coupling. No, but the mechanism of the excitation coupling is very different. In this one, we have most of the calcium. It comes from the extracellular fluid, right? Not from the sarcoplasmic reticulum. Calcium channels in the sarcolemma, it admits or allows the calcium to enter in, but they are of different types, including the voltage gated, the ligand gated, and the mechanically regulated, which basically respond to stretch. Now, the smooth muscle has no troponin. And instead, the calcium is going to bind to the protein called calmodulin, which is associated with this myosin. Let's see where this thing is. Okay, so the calmodulin, it's going to activate the enzyme myosin-like chain, the kinase, which adds a phosphate to the protein of the myosin head. Now this phosphate is going to, in addition, it activates the myosin ATPase so that the myosin can bind to the actin and then hydrolyze this ATP. Now the thick filaments, they're going to pull on the thin filaments, which in turn are going to pull on the dense bodies and the membrane plaques, and it's going to transfer that energy or that force to the plasma membrane and shorten the entire cell. Now, when a smooth muscle cell contracts, it puckers and it twists somewhat like wriggling out of a wet towel. Now, compared to a skeletal muscle, the smooth muscle is very slow to contract and relax. Now, the latent period, which you talked about earlier, between the stimulation and contraction in the smooth muscle is only 50 to 100 milliseconds, compared to 2 milliseconds in the skeletal muscle. Now, tension in the smooth muscle peaks at 0.5 seconds after stimulation, and then it declines after a period of 1 to 2 seconds. Now, smooth muscles myosin ATPase is a slow enzyme, and then it pumps the removed calcium from the cell are also slow. Now, as the calcium ions fall, the myosin's ability to hydrolyze the ATP drops, but then it does not detach from the actin immediately. Now, it has a latch bridge mechanism that's going to enable it to remain attached to the actin for a prolonged time without consuming ATP. Now, some smooth muscle often exhibit tactiness and is highly resistant to fatigue. It makes ATP aerobically, but its ATP requirement is slow. It requires 10 to 3,000 times less ATP than skeletal muscle to maintain the same amount of tension. So here we are with a relaxed, smooth muscle. Here's the plaque, here are your intermediate filaments. Here's your actin, there's that dense body, and there is the myosin, the dark purple ones. Okay, now this is in a relaxed state. Then when it contracts, here we have don't forget the filaments, the actin filament and the myosin, they don't change in length. All they do is slide over each other. Okay. Okay, so stretch alone sometimes causes smooth muscle to contract. Now, distension of the esophagus with food or the colon with fetus causes a wave contraction called peristalsis. Now, smooth muscle exhibits a reaction called stress relaxation or receptive relaxation response. Now, when it's stretched, it briefly contracts and then it resists, but then it relaxes. Now, this response, it allows hollow organs such as the urinary bladder to fill without the smooth muscle in the walls expelling the contents immediately. Now, smooth muscle is also less limited by the length tension relationships that skeletal muscle, so that even when stretched, the smooth muscle can contract forcefully, such as when emptying a full urinary bladder. Okay, so there are three features that contribute to the smooth muscle's ability to stretch extensively, and they contract powerfully. Now, it has no Z discs, so thick filaments do not butt against each other and stop the contraction. Second feature, the thick and thin filaments are not arranged in orderly sarcomeres, so have too little overlap from the cross bridging does not occur. The third reason is that the thick filaments have myosin heads along their entire length, with no bare zones. That's right. So the cross bridges can form anywhere. Now, smooth muscles, they exhibit plasticity, and that's the ability to adjust its tension to the degree of stretch. Now, organs such as the bladder can be greatly stretched, yet not become flabby when empty. Now, muscular dystrophy, we've heard of muscular dystrophy before. This is a group of hereditary diseases in which the skeletal muscles degenerate and weaken, and then are replaced with fat and fibrous scar tissue. Now, the Duchenne muscular dystrophy is caused by a sex-linked recessive trait, and 1 out of 3,500 live-born boys will experience this type of dystrophy. It's the most common form. You usually see it in males, obviously, and they're diagnosed between ages 2 and 10. The mutation in the gene for muscular protein dystrophin. Now, the actin is not linked to the sarcolemma, and the cell membranes are damaged during that contraction. So then we'll have necrosis, and of course scar tissue will result. Now, these individuals, they really live past the age of 20, and it's due to effects on the respiratory and the cardiac muscle, and unfortunately it is not a curable disorder. Alright, then we have facial scapulohumeral MD, which is autosomal dominant trait, and it affects both sexes equally. Is the one down here at the bottom, right hand side. Okay, the face and the shoulder muscles, they're kind of developed more than the pelvic muscles, so you have a lot of the bones and bones protruding out in the back. Then we have a limb girdle dystrophy, combination of several diseases of intermediate severity, and it affects the shoulders, the arms, and of course the pelvic muscles, hence why the individual is tilting in. Okay, so autoimmune diseases in which the antibodies attack the NMJ, the neuromuscular junction, and then they bind to the acetylcholine receptors together in clusters. So remember, the receptors are only supposed to accept 2 acetylcholines. Now you have a cluster of these antibodies attacking this particular receptor, and of course it's a disease in females, and you usually see it between the ages of 20 and 40. And what happens? The muscle fibers, they're going to remove these clusters of the receptors from the sarcolemma by the endocytosis. The fiber becomes less and less sensitive to the acetyl, and then it affects usually the first half of the effects usually appear in the facial muscles, where you have a drooping of the face, drooping of the eyes. They may have double vision, difficulty swallowing, and of course weakness of the limbs. And of course inability to fixate on the same point with both of the eyes, and that's called strabismus. Strabismus, sorry. some kind of treatment still have. They give you like an inhibitor that would stop the breakdown. This particular enzyme inhibitor that's going to prevent the breakdown of the acetylcholine, allowing it to stimulate the muscle longer. Now you have an immunosuppressor agent that will suppress the production of these antibodies that destroy the acetylcholine receptors. Now the thymus removal, it helps to dampen the overactive immune response that causes this myasthenia gravis. Now they use this technique called plasma pheresis. That's to remove any harmful antibodies from the blood plasma. And here's an example of the drooping eyes, eyelids. Sorry. Let's see if it's nice. Drooping eyelids and weakness in the muscular eyes, and have a movement, and they start to look upward. Okay. All right. So if you have any questions, please feel free to contact me concerning anything that we've talked about so far. Okay, I will make sure you guys have a review for your exam or for your test or quiz. Well, not for the quizzes, but for the exam. This is it for chapter 11. Continue to read and do your connect. Thank you.